I want to welcome you today to QIAGEN's webinar. My name is Joby Chesnick. I'm a Senior Global Market Manager here at QIAGEN, and I will be your host for today's webinar. The title of today's webinar is Accelerating SARS-CoV-2 Genomic Surveillance with a New High-Throughput NGS Solution. This webinar will be presented in two parts. The first part will be presented by Brian Dugan, Director of Enterprise Genomic and NGS Product Management at QIAGEN, where he will introduce an innovative new SARS-CoV-2 NGS accelerated workflow solution that offers several advantages over existing ARDIC-based NGS methods, including time savings, increased throughput, and reduction in plastic wear. In the second portion of our presentation, we'll then move to a discussion of NGS and other strategies for viral wastewater analysis with our guest researcher, Dr. Fernando Spilkey, who is Professor of Virology at Fevale University, Novo Hamburgo, Brazil, as well as coordinator of the Brazilian Network for Sequencing of SARS-CoV-2. Before we begin, please note that all webinar attendees have been muted during the webinar to avoid background distractions. However, you may use your question box at any time during the presentation to submit a question. We will wait until the very end of the full presentation to answer your questions. With that, it is my pleasure to introduce our first speaker, Brian Dugan. Brian received his MS from Georgetown University in molecular biology. Previously, Brian worked in marketing and business development positions for NGS testing, as well as in research at the Unischriver NICHD. Brian is currently a director for Enterprise Genomics and NGS Product Management at QIAGEN. Well, welcome, Brian. It's really a pleasure to have you as our first speaker today. We're looking forward to your talk.
Thank you, Joby, and thank you very much, everyone, for attending today. So today's talk, Accelerating Genomic Surveillance with a New High-Throughput NGS Solution, is our first webinar that will talk about our newly launched kit, our QIAseq Direct SARS-CoV-2 kit. This kit will help to speed genomic surveillance efforts on Illumina sequencing by being able to do things faster in the lab and put more samples on the sequencer really than typically have been able to do for the last year. So a quick legal disclaimer, QIAGEN products here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. Up-to-date licensing information is available on the QIAGEN, in the QIAGEN handbook on QIAGEN or QIAGEN.com. So today's agenda is we're going to start off by introducing the new, going for a brief overview of the pandemic, talking about different variants that are present around the world. Second, we'll introduce our new QIAseq Direct SARS-CoV-2 kit, followed by some early data that we've generated internally, and then talk about, finally, our QIAGEN Digital Insight solutions for SARS-CoV-2 data analysis. Now, at the end, we'll have a Q&A session as well with Dr. Spilkey, who has joined us to speak about the pandemic from Brazil. So QIAGEN has many facets of products that are found in throughout this pandemic. From early on, the explosion in the need for diagnostic testing. QIAGEN solutions such as isolation kits for viral RNA isolation, PCR assays, point-of-care testing assays, antibody assays, right? Those are obviously things that we have seen exponentially grow over the last year. But really, since the end of last year, there has been a global explosion in the need for epidemiological surveillance or genomic surveillance. And this has been brought on by the increasing prevalence of new SARS-CoV-2 variants that have been found to emerge around the world. Now, around the globe, while many of these what are called viral variants have emerged on essentially almost every continent, we have seen that there's absolutely a need to expand beyond just local PCR testing or local surveillance and be able to take the genomic data and share it with the world. So global epidemiology here and genomic surveillance has really taken on an unprecedented role in this pandemic. We think back to 10 years ago to the bird flu. We didn't have the resources and the sequencing and the infrastructure to do what we're able to do now. And now we're able to sequence samples, learn their mutation profiles, learn their lineage, and then share them with the world through places like GISAID or through NCBI and the SRA repository. And so the information that's being shared is sequence information, whether it's whole viral genome sequencing or whether it's sequencing of the host and the virus in mixed samples, whether it's doing wastewater sampling and looking to sequence everything that's in the wastewater for potential early detection of some of these viral variants. Sequencing has taken on an unprecedented role that we have really never seen before. And when we look at the ability to actually deliver vaccines and how we were able to do that in record time, we know that without the sequencing that we are able to do and the amount that we're able to do in that scale is that we would never have been able to bring to bear the vaccines and the treatments that have been made available in record time. So today we're going to talk specifically about genomic epidemiology. When we look at, though, as I mentioned, the different variants that have emerged around the world, really we're talking about not a strain, but slight mutations or mutations in different areas of the genome. Specifically, most interest has been focused around mutations that have been found in the spike protein, as those, right, the vaccines that have been produced by different companies around the world, whether it's an mRNA vaccine or a protein-based vaccine or some other variation of that, the spike protein is the target by which, right, those vaccines are looking to put our immune system into overdrive. And so starting at the end of last year, where sequencing became much more apparently needed because of the emergence of first the B117 and the B1351, because of the thought that those variants specifically, which became very public at the end of last year, were more transmissible or potentially more virulent. They also offered a chance for what's a lower immune response or potential what's called breakthrough. Now, when we talk about Brazil, and Dr. Spilkey will be able to talk about this in more depth, is that variants have emerged there in areas, in Brazil, such as the P1 variant and the P2 variant. But all of these are not local, right? Variants are not local because we have global travel and they have spread around the world. And so all of it's important to be able to look at this, to understand how these mutations happen, and to be able to track them. Because some of the mutations that are found in one region of the world to emerge have actually emerged all over the world, right? Because this is an RNA virus, and an RNA virus is essentially, they mutate frequently here. But as this demand has increased exponentially here in the late part of last year, what we saw is there was huge, actually, strain on the global testing system. The PCR systems had been in place for almost a year, but the sequencing infrastructure and the sequencing testing in some places around the world was very good, but in many places around the world, it hadn't been fully developed. So with a huge increase in now a new area, we saw significant amounts of delays in getting products for sequencing, whether it's a library prep, whether it's indexes, whether it's plastic plates, whether it's vials, whether it's tips. All of that has played a role, right, as being able to slow down our response. And so when we talk about the QIAseq Direct SARS-CoV-2 kit, we took all of this into our mind here about thinking about how to develop a new solution that would fit that would be able to address some of those challenges. Now, I mentioned the fact that the variants or the mutations, in the spike protein here are the ones that most people think about. But when we look across the entire viral genome of SARS-CoV-2, what you see is that there are different mutations in different genes across the virus that confer or reduce some level of advantage. They also, some of the mutations actually allow for greater zoonotic transfer. There have been cases that have been reported about transfer from humans to dogs. I think very famously, we've heard about cases of minks, right? An animal that's typically farmed, where they have become mass infected. There have been cases that also confirmed that the house cat, right, has received the zoonotic transfer here of the virus. So we know as we think about this, right, mutations in a virus, the virus is trying to mutate to confer an advantage in an evolutionary capacity here. And so as we track this, all of this is important to understand. Is the virus becoming more transmissible? Is it becoming more virulent, more deadly, or less of those things, right? All of that is pertinent information to have. But when we look in depth to say to look at the B117 and the B1351 variants of the virus, now what B117 has become the dominant mutation variant in many parts of the United States where I reside. And we see that its ability to, it is increased transmissibility, right? And more and more studies are talking about the fact that 117 actually leads to significantly increased negative outcomes. And so it's what's called a variant of concern, right? And B1351, while potentially not being as transmissible as 117, has a mutation found in the spike protein, the E484K mutation, that has been thought to really be able to create some level of antibody escape, right? What's termed as breakthrough. And so now vaccine developers are racing to be able to create new versions of vaccines that can specifically address some of these viral mutations. And so what we see here, again, is another unprecedented move where we've seen companies who have specifically mRNA vaccines are able to quickly spin up new clinical trials to test some of these things. And again, none of this would be possible without sequencing here. But you can see that when you look at the viruses and the mutations that are found here, it's not just in the spike protein, right? The signature is across all areas of the SARS-CoV-2 genome. And so it's important to understand what viral variant is present and what that lineage looks like. Because, again, we have seen other mutations actually develop. Now, from a public health perspective, most of the sequencing that has gone on globally has really been built on what is called the ARCTIC protocol. The ARCTIC network is a network of what I would say is researchers who are into looking at early pandemic response. And that group globally was able to come together in early 2020 and put together primers and a protocol for the globe to really be able to sequence SARS-CoV-2 and to do it effectively and reproducibly, right, using any type of sequencing platform. Now, the ARCTIC-based network the ARCTIC-based protocol is a great protocol overall. It's been able to generate high amounts of whole genome coverage, right? And QIAGEN has since last year had a solution that is based on the ARCTIC protocol, which is our QIAseq SARS-CoV-2 primer panel plus our QIAseq FX DNA library prep kits. This is a great protocol because it generates 400 base pair amplicons, right? So it's compatible with Oxford nanopore technology or longer read sequencing upstream, But really, when you use the higher throughput machines, such as Illumina sequencers, you pair that with our Illumina compatible library prep kit or any library prep kit, and you sequence the whole genome of the virus. But there are some challenges that have come with that workflow. The ARTIC workflow requires fragmentation or tagmentation. There are ligation-based workflows. And the workflows themselves can take actually a number of days, especially when you're doing a large number of samples. So with that, we're introducing our QIAseq Direct SARS-CoV-2 kit, which is really taking the idea of what the great workflow that ARTIC has built, right? But really, instead of it being 400 base pairs and designed for long read and compatible with short read Illumina sequencing, we've built it to specifically be designed for high throughput Illumina applications that has an innovative 4-hour total workflows that includes RT, cleanups that you need to do, and is built with overlapping amplicons that reduce the risk of mutation read dropout. It reduces hands-on time. It reduces the need for more tips, right? And it's a true single box solution. So essentially, you can guarantee the security of your supply chain here. So now I'm going to go into more depth about our new QIAseq Direct SARS-CoV-2 kit. So I mentioned some of the pain points here with the original kit. And so what we've done to be more specific is that our new QIAseq Direct SARS-CoV-2 kit has no fragmentation, ligation, or tagmentation that's necessary. It uses a small number of universal PCR-directed indexes with unique dual indices here, right? That allows you to go through this workflow extremely quickly, right? So without the need to fragment, without the need to do ligation, right? You create a huge amount of library in this reaction. Now, by consolidating this, by eliminating fragmentation, by eliminating ligation, right? We've been able to do that and to essentially to reduce. It says plastic wear, but the main bottleneck here is tips, right? The tip usage is about 45% of what the typical ARTIC or longer protocols look like when we compare it side by side. Now, again, this becomes extremely important if you, as someone testing in a lab, right, are having challenges of finding tips or getting tips. And we know this is something of whether you're doing manual pipetting or you're doing high throughput automated pipetting, that becomes a challenge. Now, mentioning the original ARTIC workflow here, I talked about the fact that it is a flexible workflow that is compatible with, an Oxford workflow, an Oxford nanopore, right, upstream of an Oxford nanopore library prep kit, right, but then it is compatible with downstream solutions for Illumina sequencing. But when you look at the primers, right, they generate 400 base pair amplicons that are typically, right, are certainly something that you can do with Illumina sequencing. But when you think about targeted applications on Illumina sequencing, you typically don't generate 400 base pair amplicons. It's easier to generate something smaller, so you don't have to fragment nearly as much. And so with that, we looked at we took the same methodology from a primer design point of view, and the algorithm designed our primers as the ARTIC network used. What we did was, instead of generating 400 base pair amplicons, we generated 250 base pair amplicons here. That smaller amplicon sizes mean that it immediately fits a little bit nicer in terms of with Illumina sequencing. That also eliminates the need for the fragmentation, right, or anything else that adds time and adds effort and work to the reaction. Now, the primers themselves here, though, are, there is some overlap in terms of the ARTIC primers, and really at the end of those long amplicons. But looking at the increasing prevalence of some of these viral variants that are emerging and the different mutations that are showing up all around the genome, we know how important it is to have essentially a safety net here. And so in some areas of the genome, we have built overlapping and staggered primers that are designed to essentially to have higher and redundant levels of coverage should an actual mutation affect an individual primer site at the 3' end. The final piece that I'll talk about here is that at launch, we have 384 unique dual indices. But within the next month, it'll have double that in terms of 768 UDIs. Now, while the amount of samples that you may want to run on, say, a large throughput sequencer such as the NovaSeq, right, may not be thousands and thousands of samples. If you're currently looking at solutions where you have bank samples and want to run this at a large scale, this gives you flexibility to essentially to drop the cost of your sequencing reaction by half and double the multiplexing. So this is true of whether you have a NextSeq, right, which can generate 400 million paired end reads, whether you have a NovaSeq S prime, or you have an S4 flow cell, right? All of this allows you to double the multiplexing that's available here. Now, from a workflow point of view, I have talked about the ARCTIC workflow, and again, QIAGEN has released our QIAseq SARS-CoV-2 primer panel, which is based on the ARCTIC workflow. And so on the bottom here, what you see is the original QIAseq SARS-CoV-2 primer panel plus FX workflow. What you see is that it is a long workflow, right? Originally, when we looked at the timing of this, we thought you'd be able to do it shorter than it was. But in real practice hands, what we have seen is that essentially it's 10 to 16 hours, depending on the number of samples that are being processed simultaneously. And so with that, right, again, there's things like multiple bead cleanups that are there, fragmentation, the enrichment steps, all of this stuff takes time that we wanted to essentially to clean up. And so with that, we've been able to do a few different things to create this 4 to 5 hour workflow with our new QIAseq Direct Kit. So as I talked about early on, is that there were some workflow challenges that became very apparent when speaking to our customers. There were challenges when it came to cDNA synthesis, when it came to enrichment. Customers asked if we could remove bead cleanups, if we could remove quantification and normalization, right? And then also asked us about primer design. And so when we got this feedback here and sequencing really exploded, we had recognized already there was a need to solve some of these challenges. And so, from all of the steps in the workflow, we have improved something. And some step here compared to, say, even our previous workflow. Our previous ARCTIC-based workflow is great. It's being used in a number of national programs, and it's been able to sequence the virus to high degree, right, to high fidelity with high viral genome coverage around the world, right? But again, there are challenges, right, in terms of being able to do it faster. Can you do it quicker, right? You have a primer design strategy that reduces some of the pain points, right, that I've now talked about. So essentially what we've done is to look at all of those steps and to try and improve somewhere to provide value to customers so they can do a faster, quicker workflow and still generate the highest quality levels of data. So now I'll talk about our QIAseq Direct SARS-CoV-2 data. So the first thing when we start looking at generating NGS libraries of SARS-CoV-2 is to talk about what are we doing here, right? So we're doing targeted whole genome sequencing, okay? And so, if you think about it, if you're coming from a human sequencing background, you think about targeted genomics, which is, taking gene panels of, primers or hybrid capture, right, and sequencing, megabases of areas of what, we have a very large 3 gigabase genome here. But SARS-CoV-2 as a genome is only 30,000 bases, right? So from a point of view of being able to get the full genome is that you can do this with a targeted base application. This doesn't require, right, a library prep kit just to do a whole genome. The samples that we're talking about are also not clean as well, right? And so most of the time, sequencing is post-diagnosis, a post-diagnostic test that is done after a PCR positive. And so those samples have isolated RNA. And so then you're taking that into the reaction, but you have background RNA. You have other things that are actually potentially there, right, that you're going to sequence. So you just, throwing a whole library prep kit or an RNA sequencing kit at it is going to give you a lot more information that you need. So you're going to do a targeted approach. And here, again, we use our primer-based approach here to do this. But if I talk about the original, our original QIAseq primer panel methodology here, again, we talked about taking this 400 base pair amplicon and pairing with a library prep. And so if you look at the bottom figure here, right, what you see here is what we like to call the hump and the peak. Now, this is good quality data that I'm showing you that's generated here. But if you're used to doing a targeted application, because we would get this question many, many times, many customers just say, is this right? Does this look good, right? And the reality is when you take a long amplicon and you have to fragment it, right? Fragmentation is not a perfect reaction. Tagmentation is not a perfect reaction, right? And so you have a range of sample that's there because you have varying amounts of this, right? And so you would get the hump and the peak here. But that would, so looking at that would mean that range would affect your on-target concentration of your on-target libraries, right? And so some samples you get really, really great library yields, really, really great on-target library yields, and some samples you would see that number dip or essentially be a little bit, sort of all over the place. And that it has to do with the nature of fragmentation. Fragmentation itself, right, can be incomplete. So by now with the QIAseq Direct SARS-CoV-2 library prep kit, is by taking the specific shorter amplicon approach that's specifically targeted, that has no fragmentation, is we're generating, right, high fidelity, focused amplicon and library sizes, right? So there's not fragmentation that's incomplete. And so what you see here is when I look at the scale of my library concentrations on the bottom, you can see here that my traces is that I have 5 times higher amounts of concentrations that I'm measuring here on the top with the new Direct SARS-CoV-2 kit compared to the old one. So you should expect that whether you're working at 100 copies or 1,000 copies, that you are going to see a significant increase in your library yields. And if you're familiar with sequencing, you know that the higher the library yields that you have, right, it's better diversity that you have, and probably the better representative that your sequencing data is going to be with that. Now, the other thing, too, to think about, as I mentioned GISAID earlier, and when you submit your GISAID data or your SARS-CoV-2 genomes to GISAID, right, there are minimum standards that are generally recommended for submission and acceptance of that. I believe it's about 20 to 25 X coverage, and I think it's 95% of the genome is the minimum standard, right, for SARS-CoV-2 genomes to be accepted to GISAID, which I believe is the case. But essentially, you need to have high quality, high amounts of coverage here, right, of the virus to be able to put into these public repositories. And that's for the best, right, because that helps everyone get a really clear picture of good work that's being done and to get a good look at, the clades of the virus in certain regions here. But when we look at this, though, we would see that, we knew we needed to have a highly uniform kit and something that's going to deliver high levels of base coverage no matter what the sequencing depth is, right? So when we look at the left side, uniformity of our new QIAseq Direct SARS-CoV-2 kit compared to our original QIAseq primer panel-based solution, what you see here with the blue bar is that, the red bar, the original primer panel delivers high amounts of uniformity at both 0.1x and 0.2x of the mean here. But when you see that there's even a substantial increase, right, with the QIAseq Direct SARS-CoV-2 kit, it's because, again, going back to the fact that the amplicons and the methodology that we took is focused in terms of amplicon and library sizes. It's specific, right, for short read sequencing, so it allows us to get better overall coverage, right, with that compatibility. Now, that also means, too, when we start looking at the base level coverage here, and both of these data sets were generated with 1 million clusters, right, and so this is high-depth sequencing here that we're talking about here, is that when you do high-depth sequencing, right, you want to make sure is that you don't have overrepresentation of certain regions. Now, this was unfortunately an area that we had already seen in our original Arctic workflow that had come out at the beginning of 2020, and we had actually made adjustments and primer changes to that in the middle of 2020 to increase low coverage regions of our primer panel. But there was still a bunch of peaks and valleys that you would see because of the 400 base pair amplicons, because of the fragmentation. But again, because of the new primer design and the effectiveness of this for short-read sequencing, what you see here is that going from the original primer panel, which both, again, showed high levels of base coverage, right, 98% at 20x coverage, but when I went up to 100x coverage, I would see that drop by about 4 or 5%. But with the new QIAseq Direct SARS-CoV-2 kit, you don't see a significant drop, right, below 99% here with these control samples. Now, this is important to show that when you're doing deep sequencing, that you expect to see uniform coverage across the genome, and I'll show that in just one second. Now, as I said, right, earlier, we're talking about looking at mutations, looking at sites where the PCR testing has gone on, but then also understanding there are some areas of the genome that have been reported as having low coverage previously with some other methodologies here. So we took a snapshot of 3 different areas of SARS-CoV-2 genome: the S gene, the N gene, and the endoribonuclease gene. The S gene, or the spike protein, is obvious why I'm showing this here. And so if you look at the red bar, which is the original QIAseq primer panel, the Arctic-based solution, what you see here is that the peaks and valleys that it comes. Now, you're still seeing high levels of coverage across the entire S gene. But the reality is, though, that the higher the highs, right, or the lower the lows can be amplified when you do higher depth sequencing or you do lower depth sequencing. And so those are always areas of concern when you start to see some of that difference. If you look at the blue bar, though, the new Direct SARS-CoV-2 kit, and you essentially draw a line right at the top of the 1,000 across, what you see is a highly uniform coverage plot, right, all across the spike protein. You don't see big dips and valleys here as compared to the 400 base pair Arctic-based amplicon solution. And that's important here, right? Because again, this is going to deliver high uniformity and high coverage, right, with the spike protein. Yeah, this is true when you look at the N gene here as well, and we chose the N gene because many diagnostic targets, specifically in the U.S., right, targeted the N gene early on, and you can see that they actually both kits perform really, really well here with the N gene. And so you know that you're going to get good coverage, that uniformity is there, whether you're using the primer panel or you're using our new direct kit. But I also called here back to the endoribonuclease gene. And so there has been a fair amount of discussion, if you've looked in publication and talking about that there are some areas of the genome, again, with Arctic-based network and even some of the QIAseq solutions that we released early in 2020, where we would see areas of dropout. And with the endoribonuclease gene in the primer panel, before we fixed this and added more primers to add enhanced coverage in the region, is that we would see this region drop, right, relative to the surrounding regions, both prior and after the endoribonuclease gene. And so the mitigation strategy we took originally was to add more primers on top of that region and to rebalance the primers from a concentration point of view. And you can see here by the red bar, all of that delivers high levels of coverage. But just to reinforce that the QIAseq Direct SARS-CoV-2 kit delivers the same type of coverage, you can see with the blue bar, essentially right below that 1,000 marker here is a straight line across here, just overall, right? There's a little bit in terms of going above and below, right, but nothing really abnormal from a statistical point of view. So all in all, you're getting highly uniform, high levels of coverage across the SARS-CoV-2 genome. Now, when I take a snapshot out, right, and look at what we'll call the 30,000 foot view here, it's very difficult to put 30,000 bases in a snapshot and be able to show it on a slide. But you can see, so this looks a little bit jagged, right? But if you were to go to IGV and to look at this, or to another genome browser, you can see here in terms of coverages, right, is that when you compare the blue bars to the red bars, right, you just see that the blue bars are much more in line with each other from a uniformity perspective. There is a little bit more variability with the red bars, so the Arctic-based solution that's there. And again, the QIAseq Direct Kit delivers its high levels of coverage all across the genome. Now, there is one thing, both the 5' end and the extreme 5' end and the extreme 3' end, right? It is essentially impossible for any primer-based solution to cover those areas, both poly A tail, right, and the first, few bases of SARS-CoV-2. And so those, when you see those dropout areas are not surprising, right, as both the, again, extreme 5' and 3' end. But as there has been no biological indication that those areas are of key significance, right, to either mutations or phylogeneity or clade calling or anything else like that, we can say that really this covers the entire rest of the genome here, except for those first 50 and last 50 bases across SARS-CoV-2. So finally, I'm going to just talk really briefly about our QIAGEN SARS-CoV-2 Insight Service. And what we are doing now is you have the ability to use a cloud-based solution here that supports multiple different solutions that are out there, the QIAseq SARS-CoV-2 primer panel, as well as the new QIAseq Direct Kit, which will be launched soon for this. And essentially what we're doing is we're taking all of the knowledge and the capabilities that we have been built on with QIAGEN CLC Genomics Workbench, and now make it a fast, accurate, and scalable solution using Amazon Web Server's global infrastructure. So it allows you to essentially be able to access this anywhere in the world. You're also able to do is to generate a whole genome analysis report. And so the great thing with this is that it allows you to have information that's specific to your organization. So if you're looking at delivering this to a national agency, whether you're looking to deliver a local agency or you're just producing this, for any number of reasons here, is you can brand this. You're able to put expected QC metrics on there, the data analysis, and then information about sequence information that's used by the workflow. And this is tied to predictions by Pangolin, and then clade analysis as well as predicted by NextClade. So all in all, right, we're enabling here a solution that allows both from the wet bench chemistry side to our digital insight service side to deliver solutions really anywhere that you are. So if I look at this from a high-level point of view, any lab now can perform NGS-based SARS-CoV-2 surveillance at scale. So you don't need to have a local installation. You certainly, if you have QIAGEN CLC Genomics Workbench, that is a phenomenal tool that we have built all of our analysis on, and you can continue to use that. However, if you are interested in bringing to scale a cloud-based surveillance solution, right, it's compatible, right, with with the FASTQ files straight off your sequencer, right, and it can help to push outputs such as PDF reports, XML reports, variant files, and many other things as well. And so, you can remain compliant with things like GDPR, but still deliver these solutions to support whatever sequencing efforts that are going on where you are located. So finally, as I close up, just to come back to QIAseq Direct SARS-CoV-2 kit, it comes in two different sizes here, so both a 96 sample and a 384 sample. The solution that we have is formatted similar to many other index solution kits with an A, B, C, D nomenclature, right? And so that each of these are all 96 unique dual indices, either format A, B, C, or D, right? And all of these can be multiplexed together. Now, coming soon, available later in Q2 will be our second set of indexes, E through H. Again, these are another set of unique dual indices that allow you to multiplex up to 768 per flow cell, per lane of a flow cell, so that you could do massive multiplexing, really, and double the amount of sequencing that you can do on say, an Illumina NovaSeq right now, where typically you can run over 3,000 samples at a time if you wanted to, but you could run over 6,000 samples by using both of these sets of UDIs as they become available. So all in all, again, this kit is made to be able to support global sequencing efforts, and we continue to see more and more news about the importance of the viral variants and the mutations that have impact on vaccines, on health, on transmissibility, on all of that, right? And so we know that it's important, and so we think that sequencing is important, so we continue to support these with our solutions. So as I close up right now, I'm being joined by Dr. Fernando Spilkey, and I think so. And Joby will come back in, and we can have a discussion and a Q&A, not only regarding my presentation, but also to talk about the pandemic in Brazil, as well as strategies Dr. Spilkey has been engaged in for looking at wastewater analysis. So thank you very much.
Thank you so much, Brian. I think that was a very interesting presentation, especially will be of interest, I think, to a lot of people who are interested in finding a solution that offers really uniform coverage to help the research. I'd like to move on now for the next portion of our presentation, and it's my pleasure to introduce Dr. Fernando Spilkey, who is a professor of virology at FEVAL University in Brazil. He's also the coordinator of the Brazilian Network for Sequencing of SARS-CoV-2, and he joins us now for an interview discussion of the current COVID pandemic in Brazil, as well as strategies for viral wastewater analysis. Dr. Spilkey's research focuses on environmental virology, SARS-CoV-2 evolution, viruses causing relevant diseases in domestic and wild animals, and emerging zoonotic viruses. Welcome, Fernando. It's great that you're able to join us today to give us your perspective on SARS-CoV-2 NGS analysis, and especially with regard to our viral wastewater analysis, which I think is becoming a very important topic. So thank you so much for joining us.
Hi, Joby and Brian. Good to be here with you and all the attendees.
Awesome. I'd like to begin this portion with really just a very general kind of question about what's happening in Brazil right now. Could you briefly describe the current COVID pandemic situation in Brazil?
Oh, the situation is still worrying. We had not controlled the situation over time since the beginning of the pandemic, but it became even worse in the last few months. And I think that it has something to do with the variants that Brian have already mentioned, but also about the type of partial controlling that we had tried during this year. The fact is that the country recorded almost 355,000 deaths already, and since the beginning of the pandemic. And just for you to have a glimpse of the size of the problem, the moving average of deaths in Brazil in the last 7 days is above 3,000 deaths per day, which is the worst moving average of deaths from the disease ever recorded in the country. So I would say that we are passing through the third peak of the disease in Brazil, and the amount of new cases and deaths in the last month were really, really high. We have now a trend towards stability in deaths and new cases, but the levels are very high. And we have these P.1, P.2, B1.1.7 in many states in Brazil, and the vaccination is going slowly, so we have a lot of work in the next few months, certainly.
Sounds like there's a lot of urgency for both research as well as testing and vaccination. Let me ask you, your research specialty is virology, and in the past your lab has performed wastewater analysis of various types of viruses. So can you tell me a little bit about the different types of viruses your lab has analyzed from wastewater and the strategies you utilize to perform your analyses?
Well, since the end of 2000s, we have worked in my laboratory with the detection of different viruses in environmental matrices, including many viruses transmitted by water that can cause gastroenteritis, hepatitis, and other diseases in human beings and in domestic animals. We have worked along all this time with adenoviruses, enteroviruses, rotavirus, hepatitis A, noroviruses, and more recently we have some part of my team working with SARS-CoV-2 detection in water and wastewater. We did here everything from work using viruses detection in environmental matrices and further sequencing those viruses to accomplish something like microbiome source tracking, trying to determine what are the sources of contamination in watersheds. And we also did a lot of virus-based risk analysis. And one of our works that was widely commended in the media was that work that we made for the Rio Olympics water venues, which were unfortunately highly contaminated by viruses. And I would say that environmental virology is an interesting area. I think that the application of the concepts of environmental virology in terms of the preemptive or the predictive surveillance that could be done is very useful in the case of SARS-CoV-2. And we have a lot of possibilities to do a better work in terms of controlling the pandemic if we have also this tool with us. So it could be a very good for defense of public health as well.
Mm-hmm. So are there any lessons that you've learned from your past wastewater analysis for that you might be able to then apply to the current SARS-CoV-2 wastewater analyses that are currently starting in your country?
Well, Job, I think that this is a very important question, as I believe that some typical challenges of detection of viruses in environmental matrices are sometimes not known or are forgotten in the analysis of SARS-CoV-2 environmental matrices, and especially in wastewater. Some characteristics of wastewater may be critical issues for that matter, and also other environmental variables may influence the results and maybe have to be taken in account even in the analysis and the interpretation of the results. Detection of viral genomes in wastewater is normally not easy as in clinical samples. Identification of virally contaminated water requires assays with sufficient sensitivity to detect very low copy numbers of viral targets. That's why I'm happy to see that the solution provided by Brian today shows that you could perform NGS even in very high CTs, which is a challenge that will be faced by those people that will try to make such an analysis in viruses that are detected in water, because normally they are in very low copy numbers. So this is one of the challenges. And there are some inner points in this analysis that requires also some caution, especially the possibility of PCR inhibitors that are there normally, because sometimes we have to do concentration of these waters, and inhibitors are concentrated downstream as well, so you have to take care of it. And there are other important matters that regards to the evaluation of environmental factors that could influence the results, like rainfalls, the percentage of sewage treated in a given region, which is normally high in developed countries and very low in poor countries. So this could be also something that could be there when evaluating the results that we have. And sometimes the levels of detection is of a given virus in the environment, and SARS-CoV-2 is more likely to behave that way, is not influenced solely by the number of cases in a given region, but also by other factors that could be involved.
Great, thank you. That's, yes, interesting. I wondered if you might be able to speak to the potential utility of using next-gen sequencing for SARS-CoV-2 wastewater analysis testing. And, you know, so people are using different methods to analyze SARS, right? I mean, some people are using PCR, some digital PCR, some next-gen sequencing. So, you know, how relevant do you think NGS is for the purpose of SARS-CoV-2 wastewater testing and analysis? And, you know, how can NGS best be utilized for this study?
Well, of course, PCR and digital PCR could be very good solutions for monitoring, but I think that due to the rising of new SARS-CoV-2 variants, as has been cited by Brian, I think that due to this matter, NGS from wastewater may be a very promising tool in the future, especially if we are able to have these low COP numbers at the PCR being sequenced by a very robust platform like this one that you showed us this morning, and that could be very useful for early detection and genomic surveillance. And this could be very important as to mount preventive measures in time, especially in highly populated areas, because this could allow you to have an alert that a given variant of concern or any variant under investigation is coming to this region. So maybe even before the clinical cases, you could send this information to the authorities, and this could be very useful for prevention of new cases. So I think that this is very promising.
Excellent. Well, thank you for that commentary. I know we all hope that the situation in Brazil improves. There are a lot of areas around the world right now that are still struggling quite a bit with SARS-CoV-2, and we appreciate your insight into the current situation there going on in your country. I would, at this point in time, like to invite our attendees to go ahead and use your question box and submit any questions that you may have thought of during the presentation. We already have a couple that have come in, and we can begin a Q&A session. And I think this first question then goes to Brian. Brian, maybe you can review this just briefly a little bit. This person asks, is the direct SARS-CoV-2 kit compatible with nanopore sequencing?
So that's a great question. So we have not tested it with nanopore sequencing, and this is a consolidated kit. So where the primer panel, the original QIAseq primer panel, is separate, so that you could use an Oxford Nanopore library prep downstream from there, right, purchased from them. This one is everything you need for Illumina sequencing. So the kit is not really set up for nanopore sequencing, and with the amplicon sizes being very focused, could it work? Potentially, yes, but it would also, and it would require all the materials from Oxford, and we don't. There's nothing that we know right now that it would work or it wouldn't work. So it's hard for me to say one way or the other, but it's not really designed for for that type of sequencing. I would recommend using the primer panel as being the primary methodology to do nanopore sequencing ahead of an Oxford library prep.
Great, thank you. Another question for Brian. What NextSeq read lengths are recommended? 2 times 150?
Yeah, so this is a standard sequencing run. So 2 by 150 is the recommended with it, yep.
Very good. Another question by our same attendee here. Is a 4-hour time to completion based on manual prep? Is there a robotic protocol?
Yeah, so that is based on a hands-on with a multi-channel pipette. So it's about 4.5 hours for 96 samples because it's essentially working in the plate. There is a robotic protocol that is coming that will be released shortly within the next month that will be able to automate it. The automated protocol itself would not have significant savings in time, right? It would be mainly to remove contamination risks and to make it hands-free. And the protocol itself should mostly be able to be almost all on board an automated platform with the workflow and the fact that it doesn't have it doesn't need a lot of things to come on and off the platform. So I think by May, there should be information regarding the automation of the platform that it's available for.
Okay, great, thank you. For Fernando, a question here. What method of filtration and viral concentration are you using for wastewater samples?
At this very moment, we are using ultracentrifugation mostly. We are not using filtration or membranes. There are all these methods available. Of course, any laboratory that comes into this will have to check what could be more useful for their reality. But in our case, we are using ultracentrifugation with very good results.
Great, thank you. Another question, I think this goes to Brian. The person asked, are any of your coverage figures from actual wastewater samples?
Yeah, so this kit was just released this week, and we were not, and so we are in the process of generating that. All the data that I showed was using control samples. Excuse me. So we expect to have another more data-rich webinar coming out in May that will have information from an acute respiratory infection as well as wastewater samples, right? That will be more in depth in terms of with CT values and everything else like that. But these were from ATCC control samples.
Okay, thank you. Another question here, is the analysis report AWS service only available when using the QIAGEN Direct Kit?
That's actually a great question. I don't believe that it is. I'm not. So the bioinformatics team at QIAGEN is different. I think they have different different SARS-CoV-2 workflows that will allow you to use an ARTIC-based method. But I'm not sure if it's compatible with multiple different types of sequencing technologies, right? So I would recommend speaking to a local QIAGEN bioinformatics rep who can give you more information about this.
Okay. Another question for Brian. How many reads do you recommend for the QIAseq Direct SARS-CoV-2 kit?
Yes, this is the $1 million or million read question, I should say. All right, so it really depends on what do you want to do. If you want to do low pass consensus read building, then it's a 30,000 base genome. So just look at it from the different parameters I think that have been talked about. If you want to do a minimum of 100,000 reads per sample, right? Say in the bottom end of that, that's really possible. You will generate an average of 3x coverage around the genome. This is certainly not good enough or deep enough to upload into GISAID, and certainly not reliable enough for, I would say, really high-quality variant calling. So I think 100,000, if it's just for some scanning, right, that's probably fine, but you shouldn't expect to get, high quality variant calling. Now, and that's 100,000 paired end reads, so total reads here of what I'm speaking about. As you move up to like 500,000x coverage, right, sort of the next rung here, now you're looking at about 15x coverage here. This is still probably below the GIS-8 cutoff in terms of the sequencing depth that you need. It's probably, that is enough to, from published indications, and actually we had a paper that was published by Loma Linda University using our original kit that showed that 500,000 reads at the bottom end was enough to build consensus reads, right? So I think at the bottom level, that's probably the minimum. Now, as you go up to the next rung, 1 million reads per sample, right? Again, this is where you expect to get about 30x coverage, or maybe a little bit higher, right, in terms of coverage around average coverage around the genome. This is sufficient, right, for consensus rebuilding, for variant calling. I wouldn't say that is ultra deep sequencing. But then the next rung is sort of, 2 million and more reads, right? Which, depending if you have a super high throughput sequencer, you may actually, you may have too much space to be able to go out and to sequence it. When you do really high depth sequencing, right, you essentially lose. I mean, as long as you have a significant amount of input in, right, if you want to do high depth sequencing and find novel clonotypes, that's possible, but that's probably going to be something from a CT with a very low CT value and a high viral load, right? There's been publications about people who have been immunocompromised, who have high viral titers, right, that they're looking for novel developing clonotypes. That could be a scenario where you would do ultra deep sequencing, right, 1 million plus read sequencing, or need to do more to be able to look at some of those clonotypes. So the answer is not very simple, but it really depends on what you want to do. And, if it's base consensus read building, I would start at 500,000 reads. If it's variant calling, 500,000 to 1 million. And then if it's, really novel clonotype identification, plus 1 million reads is probably, enough to be able to start to look at some of those things as they develop in some cases.
Very good. Thanks so much. I think we are running a few minutes over. I think we have time for one more question. It says, which is the cost for each wastewater sample for NGS? I think this goes to Fernando. In the case of Brazil, the Omics network, how much money has been invested for sequencing?
Well, nowadays we have something like $200.5 million dollars invested in our network, but we are looking for more investment for the next months. But that's mostly the amount of money that we have these days. In Brazil, one sample is with both QIAGEN and complemented by Illumina Solutions is nowadays something like $100 per sample, let's say. But that's the typical case of Brazil. And at least from what I saw from the Brian presentation, I think that we could lower the cost if we have this solution, because we have all the plasticware that is sometimes very expensive, and I think that we have chances to do much more with lower costs, maybe if we have the chance to pass through these new solutions, of course.
Great. Very good. Thank you so much. At this time, I do want to thank both our attendees as well as our two speakers, Brian and Fernando, for a terrific presentation today. There are a few questions that we were not able to answer during our Q&A, and we will get back to those attendees and with answers for you following the presentation. But again, thank you so much for your interest in this timely topic. We hope that you enjoyed today's presentation, and we invite you to come back and hear more presentations from QIAGEN. With that, this concludes today's webinar presentation. Again, thank you, and have a great rest of your day.